Hi, everyone, and welcome to our OHBM 2022 Symposium titled, What Should We Do with Neuroimaging Analytical Flexibility? My name is Angie Laird, and I'm a professor of physics at Florida International University in Miami. I want to start by thanking the program committee for giving us the opportunity to discuss this important topic. And before getting into my presentation, I want to take a moment to acknowledge that it's June 2022, and many of us are dealing with massive inequities right now because of our varying vulnerabilities. What represents low risk for some of us may be high risk for others at this point in the COVID pandemic. So I'm really thankful for OHBM to give us a hybrid meeting this year. If you are watching this virtual talk, please know that I am really grateful for your time and attention, and I look forward to a future when we can all safely meet again in person. Okay, so I'm going to start this symposium off by giving a gentle introduction to the topic of analytical flexibility, and in doing so, I'm going to address issues of reproducibility and meta-analysis as well. Let's start with reproducibility, because I think our interest in analytical flexibility is centered specifically around concerns about reproducibility. Yes, an increasing body of evidence points to some reproducibility issues in biomedical sciences and psychology and neuroimaging in particular. I've highlighted a few of my favorites right here that have illustrated the unfolding evolution of the reproducibility crisis. And a good bit of this discussion has found its way into mainstream media outlets, which definitely complicates the ongoing discussion. But, and as this very perfect tweet captures, I don't want to talk about the dead salmon anymore. What I do want to do is focus on the future and what we can do to address it moving forward. So I'd like to really reframe this as a reproducibility opportunity. Sure, we now understand that some of the things that we've done in the past haven't held up very well, but that's okay. How about we focus on working together to make our science and our community even stronger? So historically, the brain mapping community has actually always been interested in reproducibility. We just framed it a little bit differently back in the early days. I think it's important to recognize that a few groups were early pioneers in looking at consensus across neuroimaging studies, comparing activation patterns observed across multiple studies to determine where there was agreement. And when there wasn't agreement, such as in the case of the anterior cingulate cortex, they examined the experimental design across those studies, which led to these two prototypical meta-analyses on somatotopic mapping of the ACC. The first that I'm aware of was published in 1993 by Tomas Paus and colleagues. And in that paper, they examined various functional PET experiments that required hand responses, speech, and eye movements, and they mapped the variability of response types across the cingulate. And then this was followed by a similar effort in 1996. Then below that, I have listed two additional early meta-analyses. Methodologically, these were really simple, and the focus here was just to plot the coordinates across papers and qualitatively assess convergence and location. And as many of you know, since then, our interest in consensus has grown so much that we have an entire subdomain of our field that focuses on neuroimaging meta-analyses, including algorithm and tool development for both coordinate-based and image-based meta-analysis, and numerous applications relevant to, at this point, pretty much every domain of cognitive and clinical neuroscience. So my point here is that we knew pretty much from the very beginning of our field that there are limitations to any single neuroimaging study, such as our utilization of small and very frequently non-representative samples. We know that many of our imaging modalities are an indirect measure of neuronal activity. We know that our literature is full of publications that are isolated findings. And then we also know we have considerable variability due to both experimental design differences and differences in our analytic pipelines. And so for a long time now, meta-analysis has helped us say, okay, we know that there are numerous sources of variability associated with a given set of studies, but what are the consistent findings across studies? Where do our findings converge? And so now I want to focus from this point on in this symposium on this particular aspect related to variability due to analytic pipeline. Okay, so that means we're now ready to start our journey into the multiverse. And I'm hoping that there are a few Marvel MCU fans out there who are willing to join hands with Loki and Sylvie and venture past the void. Our first step into the multiverse actually began with Josh Karp's paper on the plurality of methodological worlds, which revealed to us that the choices that we make when constructing our analytic pipelines might actually be influencing some of the outcomes of our results. So in this paper, data from a single event-related fMRI experiment was analyzed using multiple different methods. The author identified 10 different analysis steps, each with 2 to 4 different strategies, which yielded 6,912 unique pipelines. And then the activation maps were corrected with 5 different thresholding approaches, resulting in nearly 35,000 maps, which is a lot. The results showed that some regional results were highly stable across pipelines. For example, there's a nice little bit in the paper that focuses on the stability of results in the right inferior frontal gyrus. But however, in general, estimates of the overall peak activation strength and statistical significance were highly variable across pipelines. All right, fast forward to 2020. Yes, 2020 gave us so many gifts, but one of the most important ones for our research community was this study, which we'll refer to again throughout the symposium as the NARPS paper, which stands for Neuroimaging Analysis, Replication, and Prediction Study. All right, in this particular paper, 70 independent teams analyzed the same fMRI data set to test the same 9 hypotheses. Notably, no 2 teams chose identical workflows, demonstrating that researchers really do have an incredible number of degrees of analytic freedom. The flexibility here resulted in sizable variation in the results of the hypothesis test. So for example, on average, across the 9 hypotheses, 20% of teams reported a result that differed from the majority of teams. And this variation was observed even for teams whose statistical maps were highly correlated at intermediate stages of the pipeline. So overall, this study showed that analytical choices have a major effect on reported results. Across teams, however, variation in the results was related to several aspects of the analysis methodology, which I'll review in the next slide. And meta-analysis was used to aggregate information across teams, and that yielded consensus in activation regions. This consensus was actually greater than what they expected based on the results of the hypothesis test. And this is a particularly important point because it demonstrates that meta-analysis really offers a solid pathway for synthesizing multiversal results. Okay, so the NARPS paper showed that even if we take the same data set and want to test the same hypotheses, then different groups are likely to end up with different answers. It's hard, if not utterly impossible, to estimate reproducibility using one single analysis pipeline. That is, the array of analytic approaches that are available to us is so vast and flexible that it is our very own analytic multiverse. Now, from the CARP and NARPS studies, we know that key stages that are more likely to influence results are spatial smoothing, multiple corrections, software packaging, and denoising. But we also know that we have a lot of choices when it comes to parcellating our brains into regions or networks using a structural functional atlas. So I also think that we need to recognize the role that parcellation plays as well. And for this particular point, I want to highlight a cool study by Ruby Kong that she's presenting at this meeting that looks at spatial correspondences across 16 group level functional atlases. This work is really going to help us make some progress in resolving some of the unknown variability associated with there being so many different atlases with competing nomenclature in use across studies. Okay, so anytime you talk about analytic flexibility with another researcher, the response is nearly always, great, it's fabulous that we know this, but what are we supposed to do about it? Or, as Morbius is asking here, what is reviewer 2 going to say about it? Okay, the first step, I think, with any large-scale community problem is to talk about this broadly throughout the community. Hence, we're having this conversation at this meeting. And I think that it's also critically important to be fully transparent in your papers about your pipelines. And this point was articulated really well in a quote from the CART paper. To mitigate the effects of this flexibility on the prevalence of false positive results, investigators should either determine analysis pipelines a priori or identify optimal pipelines using data-driven metrics. If researchers use multiple pipelines to analyze a single experiment, then the results of all pipelines should be reported, including those that yielded unfavorable results. And a tweet earlier this year caught my eye that really makes my next point. My absolute favorite thing about the brain mapping community is the community itself. We have so many amazing folks in our midst who spend their entire careers on informatics and software development, working tirelessly on the tools that are critical for advancing brain science. And what is next on their agenda is helping researchers make multiversal pipelines accessible for all of us. All right, to that point in the NARPS paper, to assess the results of 70 different analysis teams, they used image-based meta-analysis to examine the variability across each team's unthresholded statistical maps. And as described in this quote in the paper, the results show that the inconsistent results at the individual team level underlie consistent results when the results of teams are combined. So that is, if we as a research field are going to go down this complicated multiversal path, then we're going to need meta-analysis to help us aggregate results and show us where analytic flexibility gives way to consensus. In addition, NARPS also assessed variability of thresholded statistical maps. And to do that, they used coordinate-based meta-analysis using the NIMARE package. So briefly, a bit more about NIMARE. It's a Python package. It's led by Taylor Salo, whose goal is to implement a common interface across different meta-analysis algorithms and packages. NIMARE is open source. They enthusiastically welcome anyone who is interested in contributing. And NIMARE provides a standard syntax for a variety of different meta-analysis methods, interacting with both coordinate-based and image-based databases. And NIMARE has joined the ever-growing Python neuroimaging ecosystem, along with NiPipe, NiLearn, and the NeuroSynth platform. All right, so let's circle back to the original topic and revisit what I referred to as the positive framing of a reproducibility opportunity. Neuroimaging is still in the process of transitioning to a more replicable and reproducible future, but we've made progress on several fronts. Our sample size across studies continues to grow. Technologies for our analytic pipeline and their containerization are becoming more and more prevalent, and more researchers are embracing pre-registered analysis plans, and more journals are accepting submissions as registered reports. But we do have some unanswered questions that we want to work on. We know that analytic flexibility impacts outcomes, but how severe is it? And what are we actually supposed to do about it? Should we avoid it? I hope not. How do we manage it? That's a great question. Is this even computationally feasible? Is it sustainable? And how are we going to make sure that we're appropriately considering computational equity and accessibility? We're all at different locations and have a different access to resources, and we need to make sure that there's equity in how this is pursued as a field. And while I highlighted meta-analysis as a tool, we do need to work through all of the issues related to examining and summarizing multiversal results. And then finally, and so importantly, what are the implications for the publishing process? Should reviewers or editors request multiversal analyses? What are the guidelines or restrictions or limitations for doing this? So that's a lot of questions. Some of these we'll answer in the rest of the symposium, and some will just say, hey, these need more work, and we as a community need to start working on them. So I leave you with a relevant quote from He Who Remains when he's explaining the multiverse to Loki. You know you can't get to the end until you've been changed by the journey. This stuff, it needs to happen to get us in the right mindset to finish the quest. I think we're currently at a pretty messy and chaotic point in our journey of discovery about analytic flexibility. And frankly, we've got more questions than answers right now, but that's okay. We're in the right mindset now, and as a community, we're ready to tackle the hard issues. And the last quote I'll leave you with is another from He Who Remains, who said, "We're all villains here. We've all done horrible, terrible, horrific things. But now you have a chance to do them for a good reason." So look, it's really fair to say that our prior results may not be as robust and reliable as we thought at the time. And man, that BWAS paper sure is causing a lot of existential crises in our community. So yeah, we might be the villain in our own stories. But what matters now is that we know this, we collaboratively do our best now in producing good, solid, transparent research that leverages the very best resources for open and reproducible science that our field has to offer. And so learning more about that is what the rest of the symposium hopes to accomplish. And so with that, I thank you for your time and attention, and I turn it over to my colleagues First, Camille is going to give us a review of neuroimaging flexibility components and records. Then Tom is going to provide his insight regarding statistical approaches to multiverse analyses. And finally, JB will ask some really interesting questions centered around the publication process, with emphasis on how and to what extent reviewers and editors need to be considering multiversal approaches. Thanks to everyone for listening. I hope you enjoyed coming along with me on this multiversal journey, and I hope you enjoy the rest of OHBM 2022. Thank you.